What? This is a ginseng. Like the trumpet, and then you could use it in all friends if you
My kid has this giant mass on his head, and
put the ginseng.
yeah, he taught himself trigonometry since he taught my daughter.
Oh
Was
wow.
a year ahead of him. So
Okay, hello, my name is JHobbs, and I did my project. So, when I first wanted to decide what my project wanted to be about, I knew I wanted it to be about something STEM related, but I didn't know if I wanted it to be like engineering or biotechnology because I've always been interested in like engineering and stuff, but ever since AP Bio sophomore year, like biology really interested me, and like I really seemed to get it. So I thought it was something cool that I could try because I mean I knew like there's different types of engineering, but I didn't know very much about biotechnology. So I thought this project would be a cool way to like introduce myself to biotechnology and kind of decide on whether I want to pursue a future in engineering or biotechnology. So my research question was if gene therapy would be an ethical treatment to be used on patients, and I found a lot of really cool stuff about gene therapy. I thought originally that it was like a very new, controversial, like research treatment that hadn't really made it to like clinical trials yet. Like it was just something that they were trying because gene therapy is actually manipulating the patient's genes and their DNA to get like specific traits. But actually, they're having like case studies where like gene therapy is actually being used. Like for example, there was I think a 5-year-old and a 9-year-old that both had autoimmune deficiencies where their immune system was either deficient and didn't work properly or was completely absent. And they were actually able to use gene therapy by modifying some of their cells so it was able to produce the right proteins and substances to allow their immune system to start working. I thought it was really interesting how this new process where it's actually manipulating the patient's genes and DNA is actually being used right now. But I found that though it can be used positively, like in autoimmune deficiencies, it can also be used in like sports to like because like specific genes might be development and stuff. So that would be an aspect where gene therapy would have to be controlled, and also it could be used like before birth. So gene therapy could be used in the reproductive cells to change the traits of children before they're even born. So that would be another way where it would be very controversial, and they really need to figure out how to regulate this new process so it doesn't get out of hand. So people are like creating their own children, but so it can still be used to help diseases like autoimmune deficiencies and maybe even cancer. So my internship was at the Virginia Biotechnology Center in Richmond, Virginia, and it's this big complex that has over 60 biotechnology companies in it, and I interned for one of the companies in it. And it was a really cool facility. It had a shared lab, so it was like these specific labs where the companies were able to share it because it had specific equipment. So like the companies didn't have to get all the equipment they needed. They just had to, I guess, rent out their room or whatever and get their special equipment they needed. But then this other equipment they could use like this one thing that was like a pressure sterilizer, and it got stuff really hot and applied a lot of pressure so it'd be sterile. And there was like incubators and really big like microscopes and stuff. And it was a really cool facility, and I got to see a lot of it. And this is where my intern worked, or my mentor worked, and her name was Marissa Mack, and she was the assistant researcher at ENARX. And this is where she worked, and it looks kind of cluttered, but it's actually very organized, and there's a lot of pipettes and stuff, and this is where we did most of our work, and she really taught me first, like I had to go multiple times, so it was kind of segmented and like in different experiments that she was doing. But she showed me how to like grow bacteria to get DNA, grow cells, how to cut the specific parts of DNA out of the DNA, and how to paste it on the other DNA so you can get the DNA that you ultimately want. So, but it involved a lot of pipetting right there. Those are pipettes, and that was one of the main tools that she used. And it was just really cool, like learning the whole process of stuff. And here, this process is called protein electrophoresis. And well, I think this one is DNA electrophoresis because in this case, these little lines right here are each a different sample of DNA. And what it would do is it would run electricity through this gel and through this like liquid, and because the DNA has a certain charge, it would move away from the top of here down to the bottom. Strands of DNA would move quicker, and the bigger strands would move slower. So the DNA would separate, and it would separate in colors along the different lines, so you could see how big the DNA was, and that's how they use. That's what they use to identify if they cut the DNA properly. And then from here, they'd be able to actually cut the gel out of, well, cut a piece of gel out with the DNA in it, and uh, I don't know, but they would extract the DNA from that piece of gel and use it to paste it on the other DNA. And I thought that was really interesting. There's also other processes. It was called an ELISA test where they use antibodies and stuff to identify if a certain protein was present after they finished the experiment to see if the cells were creating the correct protein, so they were changing the DNA to make them produce. And there was also a transfection where they would actually use like cells, like empty cells, and they would insert the DNA into the cells. And I thought it was really interesting, being able to see like empty cells without DNA, being able to put DNA in. This machine right here was used to take pictures of this because sometimes it was kind of hard to see, and when you use ultraviolet light, so you could really distinguish different lines of DNA. So that was a pretty cool machine that you used often. This machine right here is what I'm operating right there, and it was a machine with this little tray, and you would put little samples on the tray, and it would use a laser to identify the concentration of like DNA, protein, or even like it was used to measure the color because for the ELISA test, um, it would produce a different color. Like if more of the protein was present, it would produce a darker color. So this was actually used to identify how dark the color was. So I thought that was pretty cool. And it was really fun just learning how to use the machine and learning how to interpret the numbers and figure out if it was successful or unsuccessful. My community service and legacy was having a dodgeball tournament here at Houston High School, and all the proceeds from the tournament went to, well, they're going to go to purchasing supplies to make waiting room kits, and but my supervisor was Mr. Collier, the principal here, and he really helped me through the whole process. He because it was all new to me, because there were so many people I had to contact, so many people I had to like ask for permission, I had to get to the gym, I had to get the dodgeballs, I had to pick out a date. And he really led me through the whole process of making the event. It was really fun. It was kind of a little hectic, but it was still pretty fun, and I'm really glad it worked out. Um, it wasn't this violent the whole time, trust me. But yeah, it was really fun. It was really successful thing. And here are the waiting room kits. I intend on making these, which are intended for smaller and older children. I think this one's more for adults, but I still have to purchase the materials. But these waiting room kits are going to go to ECU hospitals, so they're for like children waiting for treatment in the waiting room, so they have something to do, or families of patients that are waiting for treatment. So it's just something that kids are gonna be able to do while they're waiting for treatment. I learned about myself that I really enjoyed to learn. Like, when I was first going to the internship, I was so excited to go there and learn about all the different processes and learn like how it actually worked and how they were able to take all these like little pipette things and just like squirt what looked like water into stuff and actually turn it into like making cells with specific DNA. And it was just really cool, like especially the experimental process or the experimental design was really strict in this case because there was a lot of like you couldn't have it like cross between samples and stuff because it was really small amounts and it was really cool. But it was really cool to like learn the whole process. Advice to future rich people, I would say, is to be assertive. You gotta put yourself out there. You gotta ask for what you want, and you gotta keep asking if they don't respond. Because for my community service originally, I just kind of asked once and waited till they responded, and they didn't really respond. But what I really should have done was like kept pressing it and just finally got an answer, whether it was yes or no. I mean, it's a lot better to get an answer than just sit there for classes waiting for an answer that you might not even get. So yeah, so it definitely helped me to be assertive toward the end of the project and really get my project done. I plan to attend William and Mary, I think. Probably for biology, maybe biotechnology. I still don't know like exactly what I want to do, but I mean, we'll figure it out eventually. But any questions?
My hobby is road biking. I spend a lot of time on my bike in my spare time. So, is there anything gene therapy can do to help me be faster or climb hills better, or has that ship sailed?
I mean, it probably could.
It's okay. You can say I'm too old.
I mean, I don't know exactly like the full extent of gene therapy because it's a really complicated process and I barely scratched the surface. But I think maybe eventually it could be used as like I don't know if there's like certain proteins that like affect like muscle development or muscle growth or something, but it probably could. I don't know if it would be a controversial process.
What kind of DNA were you working with?
I forget the name of it, but it was DNA of like bacteria. For example, the bacteria I grew first was like E. coli that wasn't well, it's actually modified already that didn't have like the adverse symptoms or whatever. So, but it was actually a circle. It wasn't like a double helix like chain. It was actually just like a circle of DNA, and I think it was just weird to work with. But I forget what it was called. Yes.
Do you know what kind of immune system deficiency people have that?
No, I don't.
So you spoke about the ethical concerns. What conclusions did you come to about the ethics of using gene therapy?
I mean, I think most of the processes that gene therapy use would be ethical. I mean, there's just cases. There's just like it could be used to like change how a parent's child could like look or act or be. That just brought up controversy, especially with like the Catholic Church and like other organizations that are against kind of the of like the environment, the natural process. So, yes.
Josh, when you were working with the biotech lab, did you get the sense that they had a pretty strong sense of like this is what we can and we can't do in terms of the ethics of what they were working on?
I mean, they didn't really explain to me exactly what they were working on with like what they were trying to their ultimate goal. It wasn't like human kind of test yet. So the farthest they got was